A key part of the NAD life cycle is its inevitable consumption, whether through sirtuins, e.g., SIRT1 deacetylating proteins, PARPs repairing DNA, or CD38 producing signaling molecules like cyclic ADP ribose. Every catalytic use of NAD results in the cleavage of NAD into two fragments, one of which is nicotinamide NEM. This release of NEM is intrinsic to NAD, dependent enzyme activity, not the precursor source. As long as NAD is being turned over, which it must for sirtuins to function, NEM will accumulate. Crucially, NEM is a well-known competitive inhibitor of sirtuins and PARPs. By binding to the conserved NAD pocket, NEM prevents these enzymes from efficiently processing additional NAD, creating a classic negative feedback loop. This feedback regulation is not optional or unique to niacin. It is universal to NAD turnover because every NAD molecule, regardless of whether it originated from niacin, NR, NMN, or NEM itself, is consumed by the same set of enzymes, which produce NEM as a byproduct. Therefore, any increase in NAD flux, whether driven by NR, NMN, or niacin, inevitably raises both NAD and NEM levels. There is no escape from this biochemical reality. The more NAD consuming activity occurs, e.g., ramped up sirtuin activation, the more NEM will be produced locally and systemically, raising the potential for NEM-mediated feedback inhibition. Some NR/NMN marketing suggests they avoid NEM accumulation because they don't contain NEM directly. But this is a semantic dodge. The key issue is not whether the precursor starts as NEM, but whether the pathway leads to NAD. That is then consumed. NR and NMN are salvage intermediates in the NEM pathway. NR is phosphorylated to NMN, which is then converted to NAD. Binding nets. Once NAD is consumed, NEM emerges just the same. In fact, several rodent and cell studies show that administering NR or NMN leads to measurable increases in NEM and its downstream metabolites, e.g., methylated NEM, nicotinamide N-methyltransferase, and NMT products like 1-methylnicotinamide. This confirms that boosting NAD by any precursor results in more NEM production, and higher NEM can directly inhibit sirtuins or require the cell to process it via methylation or other elimination routes. Moreover, niacin doesn't inherently create more NEM than NR or NMN per unit of NAD. Synthesized. The Price handler pathway converts niacin to NAD through NMN and NAD, then NAD is turned over the same way as salvage-derived NAD, yielding NEM at the point of NAD consumption. Thus, the net NEM burden correlates with the degree of NAD utilization, not the precursor pathway. What really matters is whether the body can efficiently recycle or clear NEM, primarily through salvage back to NAD, via NAMPT or methylation by NNMT. Chronic, excessive NEM accumulation could indeed become a problem, but this applies equally to NR and NMN, which rely on the same salvage loop, as well as niacin, which feeds NAD. Pulls from another entry point, but converges on the same consumption degradation cycle. Interestingly, some researchers argue that strategies to enhance NEM clearance, like supporting methyl donor pathways with choline, betaine, or folate, may complement any NAD precursor regimen by preventing NEM buildup and sustaining sirtuin activity. But again, this need exists regardless of whether NAD came from niacin, NR, or NMN. Therefore, it is inaccurate and misleading to claim NR or NMN escape NEM feedback inhibition while niacin does not. All B3-derived NAD precursors lead to NAD, and all NAD turnover yields NEM, which can inhibit sirtuins if not cleared. The difference between these molecules lies in how they enter NAD, biosynthesis, Price handler versus salvage, not whether they somehow sidestep the fundamental biology of NAD consumption and NEM formation.